Hello everyone, today I will be talking about nucleic acids. So, what are nucleic acids? Nucleic acids are one or two polymer chains comprising of nucleotide monomers. These nucleic acids are formed from the elements of carbon, hydrogen, oxygen, nitrogen, and phosphorus. Each of these nucleotides consists of a pentose sugar, a nitrogenous base, and a phosphate group that are combined together through the condensation process. As you can see here, the pentose sugar is either ribose or deoxyribose, and the nitrogenous base is made out of 5 elements, which is adenine, guanine, cytosine, thymine, and uracil. So, DNA, or deoxyribonucleic acid, consists of two polynucleotide chains that are intertwined in opposite directions and form the double helix. As you can see, this is the structure of the double helix. The nitrogenous base groups and on both polynucleotide chains are matched and bound together by hydrogen bonds. The nitrogenous bases for DNA are adenine, guanine, thymine, and cytosine. Adenine will pair with thymine, while guanine will pair with cytosine. Now we will look at ribonucleic acid, or as it's called, RNA. RNA, the RNA structure is a single polynucleotide chain, which is shorter compared to DNA. As you can see here, it is only one compared to the DNA, which is two polynucleotide chain. The nitrogenous bases for RNA are adenine, guanine, cytosine, and uracil. Thymine in DNA is replaced by uracil in RNA. The three main types of RNA are messenger RNA, mRNA, ribosomal RNA, which is rRNA, and transfer RNA, which is tRNA. These three RNAs are involved in the protein synthesis process. DNA is important because it is the carrier of hereditary information, which means that it determines the characteristics of living cells and organisms. DNA also contains genetic codes carried by nitrogenous bases for synthesizing polypeptides to form protein. As you can see from this picture, the genetic code is written as a series of 3 bases that determine the sequence of amino acids in proteins to be synthesized. For example, the AUG codon base sequence adenine, uracil, and guanine on mRNA is the code for methionine amino acid. The 3 base sequence in DNA is transcribed into mRNA codons, which are then translated into the amino acid sequence to form a single polypeptide chain. Okay, so last but not least is the formation of chromosomes from DNA and proteins. So, chromosomes are formed from DNA polynucleotide chains that are wound around a protein called histone. Histones do not carry genetic information. DNA molecules combine with histone proteins to form nucleosomes, and nucleosomes are intertwined to form the chromosome structure that is inside the nucleus of the cell. All right, that's all. I hope you guys have learned something today. Thank you.